I'm Teresa Przytycka. I'm senior investigator in computational biology branch of National Library of Medicine at NIH. And I'm going to speak today about mutational signature in cancer. I've been talking about mutational signatures also 2 years ago when I was here last time. So today I'm going to make the focus a little bit differently my talk, and I will focus on the insights into DNA repair that we can gain from studying mutational signatures. The outline of my talk is like I will start by an introduction to mutational processes and to the signatures. Then I will discuss expression-based method to infer relation between DNA repair pathways and those mutational signatures. Then I will briefly say about identification of DNA repair similarity by a measure called reprint. And if there's enough time, I will also say some words towards nonlinear models for inference of mutational signatures. Okay, so a brief introduction to mutational processes. As we are all aware, DNA in every single cell is subjected to constant mutagenic processes. Those mutagenic processes can be exogenous, related to environment, such as smoking, UV light, perhaps some drugs, but also there's important component of that comes from endogenous mutagenic processes. Those are processes that naturally occur in the cell. They are not precise. They introduce some errors and naturally lead to mutation. But also in cancer, we often have deficiency of DNA repair mechanisms, and then that will be compromise the process of repairing potential DNA damage and introduce typically a very large number of mutations. The cornerstone of the mutational signature analysis is the observation that many of those mutational processes introduce very unique imprint on DNA of the, in our case, say, of cancer patient. For example, UV light is known to mutate C2T, but that mutations are context dependent. And also, so that's has led to the idea of introducing a so-called mutation category, where we will divide all the mutations into categories based on the mutations, central mutation, and also based on the context in which that mutation occurs. A very popular category, and the category that's being used in most of the mutational signatures studies, is the category of triplets, when the mutated residue is the central residue, and the two flanking residua provide the context. And with that, we defined a mutational signature as the distribution, probable distribution of mutations that, mutation categories that are associated with a given mutagenic process. And then let me also add that for a given genome, we defined exposure as the number of mutations in the genome that is attributed to a given signature. Here are some examples of different signatures that are attributed to different mutagenic processes. One is APOBEC enzyme that introduces mutations to the genome. Another here is just smoking, and yet one more is the exposed to anti-cancer drugs, for example. Okay, so just briefly, I'm not going to dwell on it. How do we infer those mutational signatures? So mutational signatures are really kind of, I would like to draw the parallel between 1 week's talks when she explained how bulk expression can be decomposed into cell types. Similarly, we can have bulk mutation, and we would like to decompose them into signatures. So there are two, again, main lines of doing that, two main approaches. One is where we do not know, assume that we do not have a prior knowledge about signatures. So this is the de novo inference. So in this, we decompose a matrix of mutation counts, and actually we should say that where each column here corresponds to the mutation category, and each row is a sample, and want to decompose this matrix into the matrix of signatures and their exposures. And typical approach would be related to non-negative matrix factorization. And then on the other side, as we also, similarly as we have in the cell type decomposition, we may assume that we actually know the signatures, and our goal is only to identify the exposures of that are of for a given genome, to identify exposure of given individuals to those signatures. And typical method here would be a quadratic programming. So I'm not going to say more about this inference of mutational signatures, just as in the case of the composition of bulk data into cell type. There is many methods, many challenges, and the methods are still being, new methods are still being proposed. Nevertheless, what we have now is a big database, if you will, of mutational signatures, and many more signatures are again being proposed as new experiments emerge. Now, why would we like to study those mutation signatures? Obviously, the one very natural answer is that it is important to understand what are mutagenic processes that influence which left imprint of a given cancer genome, because that will help us to guide diagnostic and prevention. So by uncovering association of mutational signature with environmental factors, we may be able to identify those environmental risk and limit or at least avoid exposure of individuals to those environmental risks. Also, so that's quite very natural, but also we are gaining more understanding that knowledge about this mutational signature may be predictive of the therapy outcome. In particular, some of the mutational signatures have been shown to be predictive of immunotherapy response, particularly the mutational signature associated with APOBEC has been proposed to be predictive. Smoking signature is also shown to have a predictive power. Also, mutational signature, especially ones that are associated with DNA repair, may point some other may also point out to treatment by the mechanism of epistatic interactions. So SBS3, which is a mutation that's related to homologous recombination deficiency. So the patients that have this deficiency respond well to PRP inhibitor, which kind of inhibits the alternative pathways that allow the cells with the homologous recombination deficiency to bypass that deficiency and still divide. So this is the motivation for which we would like to now look into these mutation signatures from the perspective of whether they will bring us some understanding about what is the DNA repair mechanism of the damage that is contributing to a given signatures. So just briefly, there are many known DNA repair pathways. They usually are specific to particular type of damages. For example, the one most to the right is mismatch repair pathways that allow to fix the mismatches, and then, without going into details, various types of damage activate various type of DNA repair pathways. So we'd like really to understand which pathway is activated for a given mutational signatures. And since also we know those pathways, we know the genes involved, that may be really helpful to design a therapy similar to what we've seen with signature 3 and PRP inhibitors. Okay, so the first approach that you could naturally envision would be that for many cancer patients, we have both mutation data as well as the gene expression data. So we would kind of expect that correlating exposure to a given mutational signature with some gene expression can help us to identify the genes whose expression is activated where a given signature is present. So we tried that. This is simply based on correlation between gene expression and signature exposure. We developed this method EcoSig class where where we utilize data sets, cancer data set, which has both expression and mutational signature, and therefore every patient we can identify the profile of gene expression for that patient, and for the same patient we can also identify the profile of mutation of exposure of mutational signature, and we can ask whether those two profiles correlated or not, and for picking up all the genes for which we find a significant correlation, we can cluster the data and identify the cluster of genes that have correlated with individual signatures. Now, that method was only partially successful, especially in the terms of identifying DNA repair mechanisms. So this is the result of the method applied to breast cancer data, which is perhaps characteristic, whose characteristic property is that there is quite a lot of signatures that are observed in this data. And what you immediately see is that, while we do the enrichment of those clusters that we identify using co-enrichment analysis, there's one cluster that is enriched in nucleotide excision repair pathway NER, and we can see maybe that there's some enrichment here in the signature. Doesn't matter the number, but this is a clock-like signature, signature 5, that's suggesting that there is some involvement of NER pathway in repairing this signature. But then for the double-stranded break repair, we really cannot pinpoint the signature that's really correlated. It's correlated with many different signatures. So that is known issue that is also observed in gene regulatory network, that you have a lot of a correlation, and the simple correlation methods have their limitation. So we wanted to go a little bit, borrowing the ideas from a gene regulatory network and say, what if we build a network that will contain genes and signatures, and kind of try to see what would be the information flow between genes and signatures using methods that are borrowed from the construction of gene regulatory network. And why we can do that is that, again, for each having this kind of graph with two type of nodes, we can treat it as more uniform graph by observing that with each signature we can associate activity of signature, which will be exposure of that signature in given patients. And similarly for each gene, we can associate activity of the genes, which will be simply gene expression. So we can forget what was the a particular phenotype, but we can basically what we can say is that we have genes, we have two type of genes, two type of nodes, but we have associated with them activity. And now we would like to just use the methods that are characteristic for inferring gene regulatory network and see if we can learn something more. So this is what Bayabatar Amgalan in my group did. He, could use many of the existing methods. He wanted to refine those methods. So starting with a matrix of genes and signatures and their activities, he built a regulatory network, not a regulatory network, but we call it information flow network, where for each node, be that signature or a gene, we would like to explain the activity of the gene as an optimal combination of the activity of other nodes, and having selecting a relatively small number of minimalizing the number of nodes needed to be for the explanation. So that gives us a directed graph showing the information flow between those nodes. Some of this some pairs of nodes will have just one directional edges. For other, we will have edges going from two directions. We cannot really resolve it. We apply additional step here. That was probably the only novelty of this particular inference to try to resolve the directionality. But I will not have time to go into that. So at the end, we have this directed graph of nodes, mixture of nodes that are some are genes, some are signatures. I kind of combine here, groups of nodes here into one corresponding to genes into one kind of node. And for all the incoming and outcoming edges, we do the Go enrichment analysis to understand what sort of pathway may have impact of on a given signature, and what sort of molecular pathway can be activated or deactivated by given molecular signatures. And that's been much more powerful than a simple correlation analysis, as you would expect. And for example, there is this signature 8 for which the cause was not really known, but at least we can see that is associated with downregulation of nuclear excision repair pathways. For some, just to also to confirm that this is really, we are pulling up an important contributor. We know that the DNA double-stranded break repair deficiency is associated not only with the mutation of BRCA1, but also with the downregulation, and we really pick it up. For the signature 3, the signature that is related, with the deficiency of the double-stranded break repair. Of course, double-stranded break repair is, leads to various changes on the chromosomal. By itself, it's not a process that generates single mutation, single SBS, single mutation. So there must be some other process that is being activated that during double-stranded break repair that is leads to creation of these highly mutated genes. And what we see that was likely to be activated is that during this break and during this, the cells that are impacted by the homologous recombination deficiency, we see as activity of the translation synthesis, which are, tend to generate error as they try to fix the issue. There's also, why not related to repair mechanism, but I also mentioned before that those signatures appear to be predictive of immunotherapy response, and we looked at that for a couple of situations, for a couple of cancer, and for breast cancer, what we see by this analysis that APOBEC signature is activating Tregs, and those are known to be involved immunotherapy and predictive of immunotherapy. So that's kind of suggests that the joint analysis of mutational signature and gene expression allow us to uncover at least some DNA repair mechanisms that are activated by a given signatures. The second line of attack would be, can we have this big library of signatures. For some of them, we know a lot, but for most of them, we do very little. So is there a way to find a measure of similarity between signatures so that we can have kind of guilt by association to say, okay, if those two signatures are similar by that measure, then they are likely to have the same DNA repair mechanism. So we've been thinking about this for quite some time, and just to put you into the focus is we have to really think about the signature as being the summary of two steps. One step is that some sort of DNA damage happened, right? And then this DNA damage is being repaired by the DNA repair pathways. So now we would like to somehow, and the signature is actually encompassing both steps. So we would like to find out some kind of signature or some kind of imprint that the second step is having on the signature, so that if we have two signatures which may have different DNA damage but have the same DNA repair step, so that those will have a measure of similarity measures that will suggest that this DNA repair step is potentially similar. So thinking about it, when you put it in this way, you can now see that the DNA damage does the damage, DNA repair does the repair. So if you want to figure out something about DNA repair, you should change the mutational signatures that are probability distribution over those categories into conditional probability distribution, where I would say, okay, what is the probability distribution of a given mutation subject to the fact that the mutation happened, so that there is a damage and the damage was repaired in a particular way. So that's led us to define what we call a reprint. A reprint is a conditional probability. It's a transformation of the mutational signature, and basically say what's the probability of a given mutational category of a given triplet subject to that a mutation in that particular triplet happened. So that measure certainly allows us to avoid any biases in terms of nucleotide biases in different regions of the genome. And one of the kind of motivating examples that push us to follow this idea is that there are many signatures that are associated with DNA, with mismatch repair. And here is two signatures, 6 and 26, associated with mismatch repair, and their cosine similarities are, completely, they're not similar. Yet, if we do the reprint, the cosine similarity is 0.92. So it's really, really capturing things that the signature itself cannot capture. So this is like an early study when we look, to compare, do we really see by reprint something that we couldn't see but signature? And that was, as I say, this is from 2020 when we simply clustered the small number of signatures that were known at this time using cosine similarity of signature and cosine similarity of reprint. And we could see here, focusing on mismatch repair signatures, that they form a beautiful cluster in the reprint clustering, but not in the signature clustering. Okay, so now, some years later, we have more mutational signatures, and in addition to the altered catalog of cosmic signatures, there have been some effort to come up with experimental signatures. Those experimental signatures would be either exposing the cell to a particular chemical and see what mutational profile would emerge, or say, performing crisp deletion of, say, some protein, some genes that is involved in a given DNA repair pathways. So those experimental signatures gives a little bit hint as both what is the mutational process and also what may be the repair mechanism. So now we took all the and this is work in progress. We took all those cosmic signatures, all those environmental and signatures and knockout signatures, and clustered them together using reprint. Now, with this big set of signatures, there's many of the signatures which we do understand a little bit. So for those, we expect them to have the same DNA repair mechanism, and those one are here colored by various colors, like homologous recombination deficiency is red, those aromatic environmental factor are here dark green, reactive oxygen species are blue, and so on. So you can really see that the distribution is not random, and we have like all this mismatch repair again cluster together, although not ideal anymore, and mostly because we would expect that there are some other signatures that may have the same repair mechanism which is involved in the situation as mismatch repair pathways. Okay, just comparing that this is we're gaining more by using reprint as the similarity function than just signature similarity. Those are various clusters, and we could see that with one exception, reprint cluster things that we know are supposed to be have the same repair mechanism together. So once we're having that, we hope that we can use this tree that we built and use kind of guild by association and pick up some signature for which DNA repair mechanism may not be known, or perhaps we didn't know, right? And see if it's correctly predicted. And that's indeed the case. So going back to this group of here signatures that are, most of them are environmental signatures that have been artificially generated that contain really various chemicals that are known to be in particular in cigarette smoking. You can see it nicely clustered with the cigarette, with the SBS4, which is signature associated with smoking, but something that we expected that was colored the same way, but also clustered together with the signature 8, suggesting that the signature 8 is having the same repair mechanism, it's likely to have the same repair mechanisms as all those other nodes. And that indeed is the case because, as we also seen in the previous by using expression analysis, signature 8 was associated with NER pathway, and all of those are also now associated with NER pathway. And we can go and look at other groups of signatures. For example, maybe here we have 3 nodes. We're trying to look at the tight cluster. So we have 3 nodes associated with ROS damage. And again, we look at the signature and we look at the literature, and indeed the chemical that is associated with the signature is known to produce ROS damage. And perhaps also interestingly, as we say, the mismatch repair deficiency signature cluster together, but not ideally. So what are the other guys here? And we could see that other guys is signature 1 and some other signatures for which there's a good evidence that all of them are being repaired by BER pathways. So this is, of course, this is a prediction. There's lots of challenges and presumably false positive, but at least we have some suggestion and some way to look at those, gaining finally some information from the signature other than, okay, this patient has that signature. Okay, so as a conclusion, so this repair similarity can really indicate a repair mechanism through guild by association. And I would like to say a few words about using alternative way of modeling mutagenic processes. But before I go into that, I would like to ask how much time do I have? Okay, so I'm not going to go into details, but only motivate the question. And this is actually published at Recomp and Tel System. So obviously, we now should appreciate that DNA signatures are kind of acquired by two steps. First, DNA damage, and then DNA repair. Now, if this DNA repair is being deficient, like mismatch repair deficiency, what is going to happen now is that if we have a mismatch repair deficiency, then different DNA damage will be producing different final signatures. And we postulated that there's about 6 or more mismatch repair signature in the database. So we postulated that this might be one reason for which we have so many mismatch repair signatures, is because really those signatures are really changing the second step. So rather than having this additive model here, we propose that we should have a model that will be the two-step models, where we'll have something that we call primary signatures, and then we'll have secondary signature like mismatch repair that kind of act of primary signatures. And just without going into details, with this model, we've been, at least for breast cancer, being able to reduce all the mismatch repair signature to just one secondary signatures. So in conclusion, joint analysis of mutational signature and gene expression allows us to uncover DNA repair mechanisms acting on a given signatures, with, of course, some glass is never completely full. Another way, this reprint similarity that we introduced can indicate repair mechanisms guilt by association. And also, the last kind of couple of slides that I went very briefly over, that if we would like to capture both DNA damage and DNA damage deficiency and the initial environmental type of signatures into one model, then we should look at the nonlinear model, and we had some luck in developing those nonlinear models. And I would like to acknowledge members of my group and collaborators. Especially, I would like to acknowledge Damian Wojtowicz and member of his group, Marcin Wierzbiński, who is in the audience, and Rodette Sharan from Tel Aviv University. And thank you.